The next topic is protoplast fusion. Cells without the cell wall are referred to as protoplast. These protoplasts can uptake DNA directly in presence of certain chemicals like polyethylene glycol, that is PEG. Higher concentration of polyethylene glycol causes permeabilization of plasma membrane, which allows the uptake of DNA into protoplast. Even electric signal can be used to create small holes in plasma membrane by passing the electric current. This is known as electroporation. These small apertures then help in foreign DNA uptake by protoplast. This is subsequently followed by cell wall formation and initiation of cell division. However, the production of transgenic plants by direct gene transfer to protoplast does depend on an efficient system of protoplast to plant regeneration. Gene gun or biolistic gene transfer. This is the most recent technique where one can deliver DNA of interest into a cell by loading microparticles like gold and tungsten at a high velocity. This process of delivering DNA is called bombardment. Inside the cell, DNA molecules are released from the particles and eventually this DNA gets integrated into the nuclear or organelle genome of the host cell, and the tissue explants are eventually cultured on media containing specific hormones or antibiotics to select the transformed plants. Once the gene of interest is transferred to the desired host, its stable integration needs to be established, which is done by regenerating plants. This is achieved by the tissue culture techniques. The process of tissue culture can broadly be divided into four stages. First stage, it includes the selection of the suitable explants and its transfer to nutrient medium. And the next stage is it includes proliferation of growing tissue on the multiplication medium. And third stage is it includes transfer of growing tissue to media where it can differentiate into its various parts. And the fourth stage is it includes transfer of plantlets to the natural environment. However, genes of interest can be added into two ways as sense and anti-sense orientation. To express or overexpress the gene, one can put the gene in sense orientation. However, if one wants to block the undesirable product or a step in a biochemical pathway, one can put the gene in an antisense orientation. And the next is selectable and scoreable marker or reporter gene. A gene is used to be determined if a nucleic acid sequence has been successfully inserted into an organism's DNA. Variant DNA fragment that gives some information about gene of interest. Features of an ideal reporter gene. It is easily quantifiable and relatively rapid degradation of the enzymes and high signal to noise ratio, and it should not be toxic to cells. And fifth one, it products of the reporter gene should be resistant to the chemicals used in the processing, and it assay should be sensitive and reliable. And types of reporter gene. First one, screenable or scoreable or visual marker. It will make cells containing the gene look different. There are three types of screening commonly used. First one, green fluorescent protein makes cells glow green under ultraviolet light. A specialized microscope is required to see individual cells. Yellow and red versions are also available. So scientists can look at multiple genes at once. It is commonly used to measure gene expression. And GUS assay using beta glucuronidase is an excellent method for detecting a single cell by staining it blue without using any complicated equipment. The drawback is that the cells are killed in the process. It is particularly common in plant science. Third one, blue white screen is used in both bacteria and eukaryotic cells. The bacterial lac Z gene encodes a beta galactosidase enzyme when media containing certain galactosides is added. Cells expressing the enzyme convert the X gal to a blue product and can be seen with the naked eye. The strategy is therefore to integrate the insert. Blue colonies, on the other hand, will be able to convert the X-gal and give rise to the blue precipitate because the DNA insert either did not integrate at all or not at the correct location within the plasmid. And the next one is selectable marker. It it protects the organism from a selective agent that would normally kill it or prevent its growth in a transformation reaction depending on the transformation efficiency. Only one in the several million to billion cells may take up DNA rather than checking every single cell. Scientists use a selective agent to kill all cells that do not contain the foreign DNA, leaving only the desired ones. Antibiotics are the most common selective agents in bacteria. Antibiotics are used almost exclusively. In plants, antibiotics that kills the chloroplast are often used as well, although tolerance to salts and growth inhibiting hormones is becoming more popular. In mammals, resistance to antibiotics that would kill the mitochondria is used as a selectable marker. Thank you.